So now we have 3 short talks. These are very tight. It's going to be 15 minutes, so ideally about 10 minutes for the talk, 5 minutes for questions. And our first speaker is Dr. Puneet Rawa from the University of Oslo. And the title of his talk is Understanding the Role of T-Cell Receptor Repertoire in T1D Status. So Puneet, please come on up. Welcome.
Thank you. So today I'll be talking about my ongoing work on understanding the role of T-cell repertoires in type 1 diabetes. So just to give you the brief introduction of type 1 diabetes progression. So first you have a genetic risk based on HLAs, which leads to beta cell distress and dysfunction, and then you have autoantibodies built up in your body. And once you have multiple autoantibodies, then you suffer from the person suffers from the dysglycemia, and which leads to which requires the insulin treatment, and then you have a long-standing type 1 diabetes. So the current method for diagnosis of type 1 diabetes is basically based on the autoantibody presence and the blood sugar level. So how the T-cell repertoires can help in the type 1 diabetes diagnosis? A person has an immune history, and it is all stored in your T-cell repertoires, and it can be disease, infections, or the vaccination. And then you can use the TCR repertoires to classify the disease and healthy, or you can also identify the signature for that particular disease. Here, in this case, we are using it for type 1 diabetes. So this is our data set. So we have more than 2,200 TCR repertoires, which are divided in 3 different cohorts. So the cohort 1 we mainly use for training our model and also for analyzing, and it has 4 different clinical groups where the type 1 diabetic patients, and then the first degree relatives, second degree relatives, and the control. And the cohort 2 and 3 we have used as a held out test data set. And these are our goals, which I'll discuss in these slides. So coming to the first goal is can we find a difference based on repertoire level diversity and similarity analysis. And for that we did 3 different analyses. We calculated Hill-based diversity profile for each repertoire, and then they calculated the correlation with all other repertoires. We also looked at the overlap of public clones and also the Merz-Stahon similarity index. And with all these methods, we couldn't find any clustering based on either age or high-risk HLA type or the clinical groups. So we saw that the type 1 diabetes prediction is a very complex problem, so we have to dive deeper into it. So next we looked into the HLA and CDR3 association. So you can see in the figure that there is a HLA peptide in CDR3 beta complex, and CDR3 beta is in a very close proximity to HLA. So there might be potential impact of these HLA on the CDR3, and there is a previous study by Ishikaki. It's a very comprehensive study where they have shown there is association. And we did the same analysis here to see how the variation in HLA sites affect your position-specific amino acid frequency on the CDR beta sequences. So for that, we also did multivariate multiple linear regression model, and with this we looked at the variation in each of the HLA site and then all the amino acid frequencies on the CDR3 beta sequences. Then we did a MANOVA test with a null model where we didn't consider the CDR beta frequencies, and like that we have done around 2,800 tests based on 398 HLA sites with variations, and then 70 CDR3 beta positions for length 12 to 18. And we found around 13,000 significant association. So that means there is a restriction based on HLA on CDR3 sequences. Further, to take it a step further, based on a previous study on 18,000 case control samples, they have found out that there are 3 different positions on HLA, which are linked with the 90% of the phenotypes related to type 1 diabetes risk. So we used this HLA risk score to see can we find the amino acids on the CDR3 beta sequences that are associated with type 1 diabetes based on genetic risk. And for that we did the logistic regression analysis where we take the HLA risk score as an independent variable, and then we use CDR beta frequencies as a dependent variable. And these are for each position we have found the positively and negatively associated amino acids, and the effect sizes are basically your correlation coefficient. And we have taken only for the significant p-values of less than 0.05. And combining all these different length and amino acids, we've also designed a consensus motif, which also shows a genetic risk for type 1 diabetes. And you can see the motif score is much higher for the type 1 diabetic repertoires, and that it gradually goes down for FDR, SDR, and control. And we also use this correlation coefficient we obtained that we call effect size to calculate the CDR3 risk score by summing up it for each repertoire and then calculating the average for the whole repertoire. And we found that the CDR3 risk score shows association with different clinical groups, and it also shows a very good correlation of around 0.05 with the HLA risk score. So further, we wanted to see how this HLA risk score works as a classifier, and we use it as a baseline model. And the HLA risk score was a very good predictor of the positive and negative repertoires. And we see that at cohort 2 and 3, we got the AUROC of around 0.85. And when we see the HLA risk score in a different clinical groups, we see that it is much higher for the type 1 diabetes compared to the other clinical groups. So we also wanted to see, compared to our baseline model, can we build a deep RC model which is not based on the HLAs and just using the CDR3 sequences, can we classify the repertoires? And it turns out we could not, the performance was a bit lower than the baseline model. But the interesting thing is that we obtained a motif from the deep RC. When we use this motif as a simplified version of the complex model, we again see the same association where the type 1 diabetes has a higher motif score, and then it gradually goes down. And the most interesting part here is that when we classified these motif scores based on different high-risk HLA alleles, so the DR3, DR4 are the high risk for type 1 diabetic, and we see for the repertoires which has at least one allele which is not associated with type 1 diabetes, we see more of a difference in the motif score. So motif score is much higher for the type 1 diabetic repertoires compared to others. So this analysis complements our HLA analysis, where in HLA analysis we saw that there is a very high association with the type 1 diabetes risk, but for the repertoires which doesn't have a high HLA risk, this deep RC model works better. And the same thing we also saw for the autoantibody counts. So if the person doesn't have only 0 or 1 autoantibody, the motif score is much higher for the type 1 diabetic repertoires. So to conclude this talk, so we have found that repertoire level similarity diversity analysis are not sufficient for classifying the type 1 diabetes status. And then we also observed that CDR3 sequences are restricted by the HLA alleles. And then we have identified the motif as well as the position-specific CDR3 amino acids, which are associated with type 1 diabetes risk. We found that HLA risk score is a very good predictor of type 1 diabetes, but for the repertoires which doesn't have a high-risk HLA allele, the deep RC motif can be a good predictor as well. We have done a lot more analysis, so please, you can talk to us on the post number 7 and we can discuss further. And in last, I would like to acknowledge our team in the University of Oslo and our collaborators in the University of Florida, University of Linz, and at Fred Hutch Institute, and our funding agencies. Thank you.
Wow, great job keeping on time. Amazing data. So let's see if we have some questions from Slido. And yeah, it looks like we have one. Have you looked at the specificity of the CDR3 found?
So I'm not sure what the question means. We have found motifs, and we also looked at the CDR sequences which are associated. So there are a lot more analysis, and now we are trying to combine everything together. So we will ultimately have some CDR sequences as well.
Right. So a lot of this, just to clarify, is from bulk sequencing data, correct? Where you can't necessarily know what the specificity of the receptor is. Okay, what about considering also TRBV genes instead of the CDR3 as for correlation with the HLA score?
So we have looked at the TRBV genes, and not really with the HLA score, but we couldn't find much difference there. So we have done generalized analysis, and we couldn't find difference. We also have some cluster CDR sequences and V gene combined together, and we do see their association with the HLA.
The third one, do you think that the motif classifier works better on the non-high-risk alleles because these alleles may actually confer some risk, but have just not been associated?
So, so it's not like there's no fix that this is associated, this is not. There is a gradual thing.
Yeah.
So, yeah.
Yeah, I mean, maybe this is my naive thought, but I just think it's actually very exciting that it performs better in the low-risk situation where you need something to perform better, right? In the
Yes.
high-risk situation, you already have a pretty robust classifier in the genetic risk score, right?
True.
Yeah, okay. Any other questions from the floor? No? Okay, thank you so much. That was
Thank you.
great. Okay, so our next talk is from Martin Pezou from Sorbonne University, and he will be speaking to us today about the regulatory, come on up, the regulatory T-cell receptor repertoire as an early predictor of SLE disease progression and response to therapy. Looks like we have the wrong slide deck up at the moment, or can we move it over to his talk?
Hi, thanks for the introduction. My name is Martin Pezous. I'm a PhD student at the I3 lab, and today I'm going to be talking about the relationship between the TCR repertoire and systemic lupus erythematosus patients' response to immunotherapy. So systemic lupus erythematosus, or SLE, is a systemic autoimmune disease, meaning it affects a broad range of organs. It typically affects young women, and there's currently no cure for that disease. Regarding the pathophysiology of that disease, with a focus on the adaptive immune system, that disease is characterized by the presence of autoantibodies produced by B cells. But B cells do not act alone, right? And one of their main interactors are T cells, helper T cells, which are almost always necessary for their activation, proliferation, and differentiation into plasma cells. So that's my focus. And we can further subdivide CD4 T cells into effector T cells and regulatory T cells that act as suppressors of these cells. And we know that in patients with autoimmune diseases like SLE, there's a deficit, a lack of regulation. So the I3 lab has been at the forefront of studying low-dose IL-2 as a potential immunotherapy for treating such diseases by restoring the balance between regulation and effectors. And that's because low-dose IL-2 has this interesting feature, which is that it's able to selectively make regulatory T cells proliferate and activate without having too much of an impact on effector T cells. And a recent clinical study actually showed that this is beneficial in SLE and that patients respond to the treatment. So my question is, does low-dose IL-2 have an impact on the TCR repertoire, which in turn may lead to the clinical response that we observe in patients? And actually, we hypothesized that it's not likely that the whole repertoire is linked to that biological clinical condition, but rather a small subset of the repertoire. And that's what we call a TCR signature, and that's what we're looking for. So to look for such signatures, we can leverage data that come from a clinical trial called LUPL-2. In that trial, patients were given either low-dose IL-2 or a placebo, and their response to the treatment was biologically and clinically assessed 3 months later. We have blood samples from these patients from before and during treatment at baseline, day 5, month 1, and month 3. And from these blood samples, T cells were collected and cell sorted to separate effector T cells from regulatory T cells, and I analyzed their repertoire sequencing data. So to look for such TCR signatures, we developed a pipeline for mining TCR signatures that goes as follows. We have That's the pointer, right? So we have two groups of patients, group A, group B. They're labeled according to their biological condition or clinical state. And the first thing that we do is we try to identify the most discriminant features that separate both groups. And these features are TCRs that may be aggregated at the CDR3, clonotype, or even amino acid motif level. Now, we could stop here and analyze these signatures, but we'd have no idea of how generalizable and how reliable these signatures are, which is why this whole analysis pipeline is framed within a cross-validation setting, where some of the samples are initially left out of the analysis, and they're used later for validation. So once these discriminant features are selected, we train a very simple predictive model, which is basically a logistic regression at classifying group A from group B based on the subset repertoires, subset based on these discriminant features. Once the model is fit, we then assess the performances of that model on the left out samples. So the first type of signature that we mined were treatment signatures. What you can see on this heat map are the performances of our binary classifiers in terms of Matthews correlation coefficients. So a value of 1 is a perfect model, 0 is a random model, and negative values are contradictory classifiers. So what you can see here is that from the three earliest time points, day 5 baseline and month 1, the only and most predictive repertoires are the day 5 regulatory T cell repertoires. And we were kind of expecting that, as we know, as Ancarnita said before, that day 5 corresponds to the time point at which we usually observe Treg proliferation under IL-2, low-dose IL-2 treatment. So what is shown on this heat map here is the distribution of that signature within treated patients in green and placebo patients in orange, and what you can see is that this signature is present in treated patients, and that will be interesting, especially in comparison to the next signatures. So the next type of signatures that we mined were treatment response signatures. So again, this is a heat map showing the performances of our models at various time points across cell subsets, but this time at the amino acid motif level, CDR3 level with various numbers of CDR3s selected, and at the full clonotype level. What we can see here is that we have two predictive classifiers. The first one is based on the baseline effector T cell repertoire, and the second one at the CDR3 level, right? And the second one is the day 5 regulatory T cell repertoire, but this time at the amino acid motif level. One interesting thing about these response signatures is that in comparison to the treatment signature, they seem to be almost inverted, like a ta-ta-ta. So you can see that these signatures are present in the non-responder samples. So patients that have these TCRs are very likely not to respond to the treatment. So now that we've identified such signatures, we will refine them and integrate data about some more clinical data, information about their HLA, and then go into the longitudinal analysis of these signatures, as well as try to infer and study their antigen specificity. Thank you very much for listening. I'd like to thank the whole I3 lab, both my supervisors, Victor Grive, Ancarnita, and you can come see me at my poster this afternoon. You can clap now.
That was great. So do we have any questions on Slido? So, no questions on Slido. Any questions from the floor? So I think it's still very early days with this, but it's super interesting that you see these effects, right? Do you have can you speculate at all about what you think the basis is for the T regulatory signature that you see? you think it's is it just clonal expansion or CDR3 shift among the expanded clones in some way? Like, have you tried stratifying it by expansion?
I didn't try to stratify it, but the main hypothesis would be that those, so for the treatment signatures, these are the probably the cells that were already prone to expansion.
Yeah.
As lotozyl 2 is not supposed to selectively make certain clones expand, right? It's active on all T cells, so that could be it. Now, regarding the response signatures, it's harder to interpret because they're negative signatures, right? They're non-response signatures.
Exactly.
So for the effector T cell signature, we could hypothesize that these are bad clones that lead to non-response, right? So because it's an effector T cell, right? So you could hypothesize that. But for the negative regulatory T cell signature, then I think it's harder to interpret, right? Because why would some Treg clones lead to non-response? That's something I didn't figure out yet, but hopefully we will with further analysis.
Yeah, and is the diversity of the Tregs that you see in the non-responders higher?
I'm sorry, what?
Is the diversity of the Tregs that you see in the non-responders different from the diversity in the responders?
I'm not sure. We did, I didn't do the diversity analysis. We did some diversity analyses. I'm not sure.
Okay. All right. So we have some questions in the meantime, and we have a couple minutes to go through a few. So do you have any suggestions on why the signatures are more apparent in the non-responders?
No. No, that was surprising, right? Because that feature selection step is supposed to select the most discriminant features. They have no reason to be biased towards one class. And actually, we have more responder patients than non-responder patients. So no, I don't know why they're more apparent in the non-responders.
I guess that gets back to the diversity question I was wondering about, right? I think that would be an interesting thing to look at. Another question is, how many CDR3 sequences are included to construct the signatures?
Yes.
That's interesting.
So the way we select these features is we tell the algorithm how many variables we want to select. So there's some tuning involved beforehand. So depending on the signature, that number differs. So for the treatment signature, we tried to identify around 1,000 TCRs. For the response signatures, the effector one was about 100 CDR3s. And regarding the regulatory T cell response signature, that was at the amino acid motif level. So even though that was a few hundred motifs, we have to keep in mind that these motifs actually correspond to a large number of CDR3s, right? So probably tens of thousands, which is interesting, right? Biology-wise.
Yeah. Yeah. So how many per person, roughly, did you say?
I'm sorry?
How many of those would map out per person?
I don't know. Per person?
Don't know. Yeah. Okay. Just curious. All right. Were there deletions or missing VJ combinations?
I don't know. I recently did the VJ distribution, but I'm not sure.
Okay. Any other questions from the floor? No. Okay. So thank you so much. Okay, and our final speaker for this session is Vincent van Duren from the University of Antwerp, and the title of Vincent's talk is Decoding the T-Cell Receptor Repertoire in Chronic Autoimmune Arthritis. Vincent, all yours.
Right. Okay. So hi, I'm Vincent van Duren. I'm a PhD student at the University of Antwerp. And my research is situated between two research groups, immunoinformatic research group, but also a more clinical group that is led by a pediatric rheumatologist. And he sees a lot of different patients with different forms of autoimmune arthritis, which is a diverse group of conditions with different disease forms, some of them sharing an etiology. But one thing is clear, that there is a role of T cells, so there is a formation of autoreactive T cells that in many cases are driving this disease. And there have been many interesting studies, but mostly the molecular mechanisms and the exact T cells and T cell receptors that drive this disease are still unknown, as well as their antigenic targets. So as our main objective, we said, okay, from these different groups of patients, can we characterize TCR signatures that are shared between different patients and can delineate patient subgroups? And can we in this way identify possibly a disease-associated TCRs? We don't have the biggest discovery cohorts, so we have to be smart, and a large part of this analysis was using all the publicly available data and integrating it and use this as a discovery cohort for these TCRs. So this involved finding public data, which is not easy, and I highly respect all the efforts to do standardization from the R community. So I went through literature and looked at all the publicly available data and parsed it. In the end, I have I compiled and curated a database with around 10 studies across almost 100 patients containing 300 repertoires. And you can see that this is across different arthritis subgroups, but also across different compartments. So the synovial fluid, peripheral blood, and a couple of synovial tissue samples, and also across different subsets that were sorted out. And if we want to do an analysis on this, we have to appreciate, we have to know that there is a huge technical bias, there are huge biases across these different data sets, there are so many decisions that are made that influence what your data looks like, and this includes both single cell and bulk sequencing data that are sequenced with different methods and so on. So to first characterize this, I briefly looked at the frequencies of the different V genes within the data sets, and you can see in this principal component 1 and 2 that it clearly separates out the sequencing methodology used, for example, for the single cell versus the adaptive immune-seq data. Also, the repertoires that were derived from the same patients are connected by edges, which is, you can see that they also group together, which is logical. But if we want to find a method that can, I first want to highlight that even though there is this huge technical bias that is present, it is not impossible to find meaningful biological signatures within this data. For example, if we use PC1 and PC2 instead, we see that this PC2 kind of separates out all the CD8 repertoires from the different studies versus the CD4 and the Tregs that are in there. With in the middle, this gray part, the unsorted repertoires. So there are signatures that we can capture that are shared across the different data sets and that are biologically meaningful, but we have to find a way to get into this and to break this down and overcome this technical bias. What I decided to look at, because of this bias, the normal methods wouldn't really work, I tried to first condense it and break it down into, okay, but which TCRs from each patient are interesting, which TCRs are biologically meaningful, and for this I looked at what I call convergence events, but you can also look at, you can also call it coincidences, and that is if you see TCR clonotypes that arise independently, so across different patients or within the same patient, but they are different in their nucleotide sequence, and they are similar. Right? So similar TCRs that arise independently, if this occurs a lot, and if you see a lot of them, this must be interesting. With this, keeping this framework in mind, we are specifically looking at TCRs that are more convergent in the autoimmune arthritis patients compared to the controls or compared to a normal patient, and we were trying to assess whether there are patterns that are delineating subgroups, and we could actually find it in this way. This method is not new. There are many people that have already done something similar. But for this specific use case, I developed a framework that found these patterns of convergent recombination, but then kept in mind that, well, used a more flexible distance metric. So I used TCR distance metric. So this also included the V gene, kept in mind or controlled for the technical bias that are in the repertoires, and thus did this at a scalable, computationally tractable rate, or in that way. So what we exactly are doing is we have a query TCR, And we are looking at all the very similar TCRs in TCR space. And within this group of TCRs, we are looking, okay, how many of these are now arthritis related and how many of these similar TCRs are from healthy control patients. And these convergent groups, we compare not to the full data set, but to like a larger radius, a radius that is large enough to remove, to go away from epitope specificity, but still look at a smaller subset and in that way remove the bias that is present across repertoires. And we tested the enrichment for like every TCR available in the database, selected the ones that are significant, and of course, for example, if this one is significant, the one next to it will probably also be significant, so you can do clustering and find these dense clusters of convergent TCRs across autoimmune arthritis. This is a slide that shows for each cluster how convergent it is, so compared to the background, how much more convergent events do we see in arthritis versus controls, and also how public it is, so how many patients share this cluster. And for example, what we can see is this very nice GLY blah 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 motif that has already been described previously and also highlighted in the previous talks that this is a perfect target for immunotherapy. But there are also other TCRs in there. For example, looking at this one is shared across different patients, across different studies, but most of these patients have OGA or OGA-PAGA, so they are enriched in ANA-positive arthritis patients. And then we decided, okay, we're looking at these convergent clusters, let's now look at the clusters that are differentially abundant in a specific arthritis subtype. So each cluster is a dot again, and we are looking at the enrichment of TCRs coming from the ANA-positive patients within our dataset. And as a validation, we said, okay, let's now look at our own data and see the overlap between these clusters. Sorry. So we are also sequencing many patients, both using single cell and bulk sequencing, and we noticed that for these ANA-positive enriched convergence clusters, they are present across all the patients that are also ANA-positive in our cohort. So this is mostly across the Treg and the CD4 compartments, both in single cell and bulk sequencing. All patients have one or more TCRs from these convergence clusters in independent dataset. We also noticed that they are not only like present, but also more expanded in our ANA-positive patients versus the other groups. This pattern is not completely unique. There are other patients that have TCRs from these clusters, but there is at least some statistical power for this. We also see for other subgroups, for example, for Lyme arthritis patients, that we find the same pattern. So we select Lyme arthritis clusters and then look in our independent data set, and we also see that these patients have TCRs from the Lyme arthritis clusters, which are not often present in other arthritis types. So in general, I want to kind of summarize it by compiling publicly available data is very challenging. It takes a lot of time and effort, but it can also be rewarding because it enables us to overcome the sparsity that is present in these TCR repertoires, bundle them together, and find patterns that weren't even described before in the original studies. Our computational framework that we used allows us to overcome these technical biases, look at interesting TCRs from a convergence standpoint, and then we showed that they are novel, sometimes already described, sometimes novel, can be used to delineate patient subgroups, and can be validated in independent datasets. Now, we answered some questions, but we have a lot more of other questions. For example, what is the phenotype and the role of these T cells in these patients? What is their epitope specificity? And can we confirm that they are self-reactive? So I still have a lot of more work to do, but I also have a few more years of PhD to do. So thank you for listening.
Thank you for that great talk. It was really nice. So starting with some of the Slido questions, how was the convergence log ratio defined?
Yeah, basically it's the log ratio of within a convergent cluster, what is the ratio of convergence from the arthritis patients divided by the convergence within the control patients.
Okay, second question. Could these convergence clusters of TCRs be compiled and used as a reference for other studies?
Definitely, I think it's very interesting because it allows you to, it's a strategy that's quite common. If you have something very sparse and you have groups, you look at it per group instead of doing it point by point, and it helps you to overcome the noise and get something that can be statistically significant. So I think it's very interesting.
Yeah, and so I'm just going to throw in one of my own questions really quick. So you saw some clusters in the ANA positive patients with arthritis. Do you also see similar clusters in ANA positive lupus patients or other ANA positive individuals?
Yeah, I don't have any I didn't look at lupus patients, so this is something we could check.
I mean, and you could, of course, further stratify by their arthritis phenotype and things like that, right? Yeah, that'd be super interesting, I think. Okay, another question. Building off of the last talk, will you look at the significantly depleted clusters in your groups?
I also looked into this. For the clusters that we find, it's mostly they are depleted because they are enriched in another group. So you see, if you saw the volcano plot, it goes both ways, but the clusters that are on the other side are, for example, purely Lyme arthritis clusters, and that's why they are depleted. So I don't think that patterns there are as meaningful as they were in the last talk.
Okay, great. Any questions from the room here? Going once. Slido, anyone? Okay, so thank you very much for a great talk, and I want to take a moment to thank all the speakers from the first session for their outstanding presentations.